Hello, everyone, and thanks for joining us for episode 712 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and in this episode, I am excited to share our conversation with Dr. Lynne Maquat, originally released on August 21st of 2017. Listeners, Lynne is a renowned scientist studying the molecular basis of human diseases, and in particular, the mechanisms of mRNA decay. She discovered nonsense-mediated mRNA decay in 1981, and she has been studying cellular processes involved in causing about 1/3 of all inherited genetic diseases, as well as about 1/3 of all acquired diseases such as cancer. And in our conversation, Lynne shares some excellent stories from her life and science. So get ready to get inspired in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of the People Behind the Science podcast. Listeners, today I am thrilled to introduce you all to Dr. Lynne Maquat. So, Lynne, thank you so much for joining us on the program. How are you today?
I'm well, thank you. How are you, Marie?
I'm doing quite well. Thank you for asking. We're excited to chat more about you and your work today. But first, I want to take a moment to introduce you to our listeners. So listeners, Lynne is the J. Lowell Orbison Distinguished Alumni Endowed Professor in the Department of Biochemistry and Biophysics, as well as Professor within the Cancer Center and Director of the Center for RNA Biology at the University of Rochester. She received her bachelor's degree in biology from the University of Connecticut and her PhD in biochemistry from the University of Wisconsin-Madison. Lynne then conducted postdoctoral research at the University of Wisconsin-Madison, and she worked as a faculty member at the Roswell Park Cancer Institute for 19 years before joining the faculty at the University of Rochester. Now, Lynne has received numerous awards and honors during her career, including the International RNA Society Lifetime Achievement in Science Award, the Canada Gairdner International Award, the William Rose Award from the American Society of Biochemistry and Molecular Biology, the Athena Award from the Women's Council of the Rochester Business Alliance, a Merit Award from the NIH, the Presidential Diversity Award from the University of Rochester, the RNA Society Lifetime Achievement Award in Service, as well as many others. Lynne was also named a member of the U.S. National Academy of Sciences, a Fellow of the American Association for the Advancement of Science, a Fellow of the American Academy of Arts and Sciences, and a Batcheva de Rothschild Fellow of the Israel Academy of Sciences and Humanities. And listeners, Lynne is here with us today to tell us about her life and science. So Lynne, today we want to know more about your research, but also more about you. So can you start by telling us what you like to do when you're not doing science?
Well, my husband accuses me of always doing science in some part of my brain somewhere, no matter where I am and what I'm doing. But what I like to do is I have a dog. I love dogs, so I love walking my dog. Rochester has lots of lovely parks and woods. And I like exercising, so I practice a little yoga and I work out with weights and do some cardio, try and keep the mental molecules good in my brain. And all of that seems to be working pretty well. Also helps me sleep better.
Fantastic. So you mentioned you're a dog person. I am as well. What kind of dog do you have?
I have a lab. Her name is Leah, and she's lovely.
Aw, I love it. Does she ever come to the lab with you?
Oh my gosh, I'm the second scheduler in terms of my husband first takes care of her, and when I come home, it's another play date for her.
That's fantastic. Well, great to get a window into your life outside of science, Lynne. But I do want to chat about your research next. I know you're working on some amazing things there in your research group. So can you tell us how you describe your work to someone who might not be familiar with your field?
My lab works on human diseases and what causes diseases in our cells. We have studied diseases not only to understand the causes of diseases and how our cells function normally, but ultimately with the idea of developing therapies. And I'm happy to say, after having worked on a particular process that typifies a third of all inherited diseases like cystic fibrosis and Duchenne muscular dystrophy, as well as a third of all acquired diseases, and cancer is an acquired disease. we are at the point of developing therapeutics after more than 30 years of working.
So this is a really exciting time then to be in your field, isn't it?
It is.
Fantastic. Well, we'll chat more about your research projects as we go through our interview today, Lynne. But I do want to take a moment and talk about motivation. I think it can be so hard to juggle all the different things that you're doing as a scientist. And I know you've been traveling quite a bit lately. So can you take a moment and tell us what motivates you? Do you have a quote or a saying or a force or something that you have in your head that keeps you going every day?
Well, I say a lot of things. I think what we say to ourselves, no matter what field we're in, is really important. And I'm an introvert. I think a lot of scientists are, because we like to think about things and we do that in our heads. But being an introvert, when I was younger especially, I was very shy. And so what I realized that typifies me is if I really believe in what I'm doing, then it's so much easier to do it. And even if I'm afraid, when I was young especially, I entered the field of public presentations and discovered things that were not expected. So, of course, that made me kind of stick out of the norm, which is a good thing, but also comes with its own challenges. I realized that believing in what I was doing was important and helped me through times that were challenging for me, especially being an introvert.
Certainly. And I think you're absolutely right here that a lot of scientists do tend to be introverts because these are the people who are absolutely wrapped up in their own heads, their own ideas and thinking about these things. And I know I'm an introvert myself. So was this something that you struggled to overcome as you had to embrace a lot of the public speaking and things that you do as a scientist?
I did. And it's not like I can't act like an extrovert, but I can't do it all day. Actually, it's funny that I say that because when I am asked to give seminars or consult at a place, I am active from the beginning of the day past dinner. But it's so much fun and it's so much mental engagement that my introverted nature kind of disappears. And even speaking in front of groups, it can disappear now because I get so wrapped up in what we're doing. Mm-hmm. And I think believing in what you're doing helps you step away from what other people are thinking and just engaging completely in the process of either speaking or talking with other people about their work or my lab's work. So it helps one get past the fear and get into the excitement.
Absolutely. Well, it's fantastic to hear a little bit more about you and what keeps you motivated. But I want to hear next about some of the people who might have motivated or inspired you. So, Lynne, do you have any role models or maybe inspirational figures that really you looked up to throughout your career as a scientist or maybe even continue to do now?
I certainly have role models and people who have inspired me. But I should put this in a context because I'm old enough now to have gone to graduate school when there were no female faculty in the department I got my PhD. And in fact, it wasn't uncommon for some of the male faculty members to think that women didn't belong in graduate school, that it was kind of a waste of time to train women who ultimately were not going to use science as part of a career. Sad to say, it still is true that many women, compared to men, do not use a PhD degree in science in the capacity of having a career. But all of that said, and getting past all of that, when I became a postdoc, especially, and then one gets out more into the International Science Forum, I could see that there were men and women who were particularly supportive of me and also highly successful women who were of special interest to me as role models because they had it even more difficult when they were beginning their scientific career. And I found that inspirational. We all have to find our own way, and each of us who are successful have found our own way. Everyone has their own story, and in fact, I have initiated something here at the University of Rochester called Graduate Women in Science to deal with the fact that fewer women than men use a PhD degree and go on further in science as a career.
Sure.
And some of these women who I began to know when I was younger have come and spoken to us. So I've actually heard their personal story. Everyone has a different story, and it helps young scientists, both men and women, realize that nothing in life that's worth doing is without struggle. Life is difficult, and we make the best decisions that we can at the time. But what is key is perseverance, believing in what you're doing and perseverance.
I like it. Well, I think those are two traits that scientists really must have to be successful. You can't be giving up, and you really do have to be excited about and believing about the work that you're doing. And we chatted about some of these mentors, some of these people who really helped you get through. Were there any particular traits that you noticed in these highly successful women scientists that you tried to pick up as you were going through?
There were traits. It's important to be taken seriously. I always wanted to be known for what was in my head than what I was wearing, for example. Mm-hmm. And I think being taken seriously and contributing in a way where people can learn to respect not only your science, but your opinions about basically any decisions that have to be made, I think is key. So I learned from older, successful scientists how to present oneself in a way that is credible and solid.
Absolutely. Well, I'm curious, Lynne, to hear about where your journey began. You mentioned that you were maybe a little bit of a shy, introverted kid. So how did you first become interested in science?
Well, I grew up in the country in Connecticut, and I always loved nature. I knew the names of the birds, and I knew the names of the plants. So I was good at a lot of things. I was fluent in Spanish when I was young. Of course, I forget everything now, but I also was very good writing in English. I loved math. But when I was an undergraduate, and I'm the first person in my family to go to a four-year college, so I went to the University of Connecticut, which was a state school, and I got into a cell biology lab when I was a sophomore and learned about what happens in our cells when proteins are made. And this was my first taste of molecular biology, what molecules in our cells do to make another molecule, protein. And I thought it was so cool. So I kind of got bitten at that point. I was very shy, and I wanted to work in this professor's lab. And I don't know where I got that idea in my head because I didn't know anybody who worked in a lab as an undergraduate.
Really?
But I went up to him, and I asked, his name was Stu, and I asked Dr. Haywood if I could work in a lab. And he looked at me, and he said, "A lab?" And so I kind of squeaked out, "Your lab," because I thought I was completely out of line. So he looked at me and he said, "Sure."
Oh, wow.
So I spent the summer working in his lab, which was very intimidating, but still I thought it was really interesting and cool.
I love this story. I think sometimes you just have to ask is the moral there. So listeners, keep that in mind. You never know.
Well, I went to a large school, and it's true that all you have to do is ask. And in fact, as a professor, I know that too now. You would be surprised how receptive professors are, just waiting for someone to come up and ask, whether it be, "I don't understand this part of the lecture," or whether it be, "Can you help me with my research project?" or "May I work in your lab?"
So then how did you decide to go on to do a PhD?
Well, I knew I wanted to get advanced training, and I ruled out medical school very early on because I saw a bone marrow aspirate from the chest, basically, before they used to do them in the back of the body, the iliac crest. And it totally freaked me out. I was volunteering. And then I went into a lab that was a patient's room that after I delivered the little magazine, so I'm only 15 years old, I closed the door and realized it was quarantine. And I was afraid to tell anyone because I thought, okay, I'm going to die right like that night. So between all of these traumatic experiences, and there were many others, I decided that the hospital probably wasn't a good environment for me. And liking research so much, it just seemed natural to go to graduate school.
Gotcha. So can you tell us a little bit about what your experiences were like at the University of Wisconsin-Madison?
I started out in a laboratory working on bacteria. And as an undergraduate, I had worked on an organism that was much closer to humans. And I always wanted to work on human disease. It wasn't that I wanted to work on bacteria. So this is kind of a life lesson. Just because you think you want to do something, but you end up having to do, or it just happens that you end up doing something else, that may not necessarily be a bad thing. And I'm not saying we should not be proactive in our lives, but I ended up working on bacteria, which turned out to be very helpful in a number of reasons. First, they grow really fast, unlike our cells. And so I got my PhD in 4 years. And secondly, we use bacteria as a tool to express human genes. So when I went on to the next phase of training, which is called a postdoc, for postdoctoral training, and I did that at the Maquat Laboratory for Cancer Research, I knew exactly how to propagate human genes in bacteria, I had very good techniques. And I was horrified at those in the lab who hadn't had the background in working with bacteria. They didn't have nearly what I thought were the techniques to have at hand to make sure you were growing a single DNA, for example. Working with single clones so that all of the bacteria that you're working with are exactly the same.
Oh, gotcha. Interesting. So now I know you spent a lot of time as well at the Roswell Park Cancer Institute. Can you tell us what was it like there?
Roswell Park, unlike what most people think, is not a park. Roswell Park was one of the first cancer surgeons. And I was in a department called Human Genetics that was later changed to Cancer Genetics. Roswell Park is the oldest comprehensive cancer center in the United States. And while I didn't work directly on cancer, I continued my work on human diseases. And it actually was a good environment from the perspective that my chairperson was heavily involved in mapping human genes. So we had a lot of resources that were available to my lab that were constructive. We did have an academic program as well. So I trained graduate students as well as postdocs in my lab. But I missed being in a truly academic environment. So Rochester has the medical school, and I'm in the medical school. And across the street, there's the College of Arts, Sciences, and Engineering. I missed at Roswell not having that College of Arts, Sciences, and Engineering. I missed interacting with people who are majoring in history. We also have the Eastman School of Music here, and a lot of our friends are from the Eastman School of Music. So the attraction to Rochester was not only that, having both campuses together, but Rochester has a very strong history in RNA biology, and I'm an RNA biologist who studies human disease. So Rochester was a place that I visited every 5 years when I was in Buffalo, Roswell Park. And when they asked me to consider, and I didn't have to be a chairperson of a department, I looked.
Excellent. So now that you are there at the University of Rochester, like I said, I know you have so many exciting things going on in the laboratory. Is there one project, Lynne, that really stands out in your mind right now as one that you are super excited about that you want to tell us a little bit more detail about?
Sure. We are working on a lot of things. One of the things I can talk about is Fragile X, which is a form of intellectual disability and autism. And the mechanism that I'm most known for working on and discovering is abnormal in patients with Fragile X. And we would like to understand how that is and what can be done about it. It turns out the mechanism is hyperactive. And we believe this hyperactivity contributes to the problem in neurotransmission that can plague individuals with this quite common syndrome, actually. So we're heavily involved in developing the tools that we need to study this using patient samples and also working on the mechanism in cells that we can manipulate to recapitulate what happens in the patients.
Gotcha. Now, is this mechanism in Fragile X similar to, or is it quite different from the mechanisms in other diseases?
Fragile X isn't as well understood as we would like. It is different than other diseases in the sense that there's primarily one protein that is defective, but this protein interacts with a bunch of others, and they too can be defective or can be defective instead. And as far as I know, that doesn't typify other diseases. Mm-hmm. But the behavior of the patients, the spectrum in terms of behavior or the disability of the patients can vary. And that's something else that we're interested in understanding.
Gotcha. Now, for listeners out there who might not be familiar with what Fragile X maybe looks like, can you describe what some of the symptoms are that you might see in a patient?
I think the key is intellectual disability, and there's also the potential to be autistic.
Gotcha. Well, then, wonderful to hear more about your work, and really exciting to hear that not only are you starting to tease apart the mechanisms for these debilitating diseases, but also starting to move into that area of potentially being able to develop therapeutics. That's awesome.
Thank you.
Sure. So now we talked about some of the challenges that you've encountered as a woman in science, particularly in not having some of those role models along the way. And I think there are a lot of different challenges in the life of a scientist, and we do like to highlight them on our program because I think it's really important for everyone out there to get a realistic view of what life is like as a scientist. So Lynne, do you have another story of a challenge or a failure or something you really struggled with that you can share with our listeners today?
Sure. I think everybody does who's been professionally active for a long time. Well, how is my research lab financially supportive? That's an interesting question that listeners may not be aware of, and it's through taxpayers. Taxpayers contribute to the National Institutes of Health. And most of my funding, not all of it, but most of my funding comes through the National Institutes of Health. How do I get funded? I get funded by writing very complex grant proposals that they detail my accomplishments during the last period of funding and what I propose to do during the next period of funding. These grants, in my case, span 4 years, except for my merit award, which can go as long as 10 years, but not without review. After writing a grant, the grant goes for peer review. I've served on study section. I've chaired study section. So these are groups of people who already hold NIH funds to support their own lab's research and who have expertise in the topics that are under review. And it's a very rigorous process. And I have to say, the NIH budget is not what it used to be. Mm-hmm. And it is more difficult to get funded. I've been fortunate. I've always been funded, and I've always had, at least for the last 25 years, 2 NIH grants. But we're talking about challenges. It's challenging to write a grant. It's challenging to have to explain and justify and project the kind of science that we've done and that we will do. So how is that able to be judged? That's by publishing. And publishing is the key to life as a scientist. That also goes through peer review. And it is difficult to publish. That involves submitting a paper. And papers these days, they can take years to be written.
Mm-hmm. I feel like the bar just keeps rising. Yeah.
Yes, the bar keeps rising. That's not necessarily a bad thing because it always selects for the best, but it's a challenge for everyone. So perseverance in those cases are key. And don't forget, who are the people at the bench doing the work for the grants and for the papers? It's people I'm training. It's my graduate students. It's the postdocs in the lab. And these publications matter not only because they provide money for reagents and their salaries, but it helps them go on to the next step, because they're able to do the research and publish. And then if they're a student, go on for postdoctoral training, or if they're postdocs, most postdocs in my lab want their own independent faculty position, usually in academics, sometimes in industry. They need to publish. So it's challenging. I'm not saying it's a bad thing, and I don't know of another way to review science other than peer review. It's an imperfect system, making it challenging.
Definitely. Now, is there a particular thing that you think has contributed to so much of your success in writing papers and getting grants? Do you have a secret you can share with our listeners?
Well, I think one key is being creative. So thinking critically, what do you know and what do you hypothesize? There's a big difference between the two. And under what conditions do you know what you know? Don't overinterpret your data and think creatively. And that's hard. And that's what I try and train people. I somehow have that, which is really exciting for me, but also, I think I look normal, but sometimes probably. So my husband loves me, but he sometimes thinks that I'm thinking about things that other people don't have to think about and can live a full and complete life without doing. Mm-hmm. So thinking creatively and critically, I think, are key.
Absolutely. Well, I love that you brought in this creativity, because I think a lot of people take this for granted in science because they think science, everything's kind of cut dry. People have a certain mentality about how you should be thinking in science. But I think this creativity is key to coming up with exciting and innovative and new things that people haven't tried before.
That's right. Or thought about.
Absolutely. So, Lynne, we don't just want to talk about the challenges. We would love to hear about some of your success stories next. So is there a particular favorite story of success that you want to share with us, whether it was a big win or even just a small victory that meant a lot to you?
Well, it's a process. Science and life are processes. And I've had lots of joys and lots of excitement and lots of discoveries, as well as lots of challenges. And that's where the perseverance comes in. Yeah, we've discovered a lot of things that people hadn't anticipated and are really interesting. So when I was a graduate student, we tended to study one thing a cell does in the test tube. And you can do that. But by working with cells and doing the kinds of experiments we've done, we realize that while you can study individual things the cell does in a vacuum, that's not how they happen in the cell. And when you think about that, how one step predisposes and can influence subsequent steps, even if they occur in a totally different place in the cell, it makes sense because that's how a cell can engage a quality control over what's going on. So if step number 1 isn't right, step number 2 can detect that there's a problem with what happened in step number 1 and eliminate the problem. And that's some of the things that we've discovered. And no one understood that step number 2 could see, and I'm using the word see
loosely, yeah.
Cells don't see, but could tell and have a way to figure out whether step 1 was okay, in which case the little train would carry on down the track, or there should be a block and degradation. Get rid of that problem, which could be toxic to cells. So that was really cool. And this concept has really expanded in terms of what the cell does in terms of checks and balances. So that's very exciting to me.
Absolutely. So the first time you saw evidence of this phenomenon, this almost surveillance within cells, checking on these different steps that are maybe happening before the one you're interested in, what was going through your mind?
So I realized it, and the challenge was getting other people to believe it.
Right, because there's all this evidence that says otherwise from looking at steps in isolation, yeah.
Yeah, we're not seeing this. And what I realized was very important was to not overinterpret what we were seeing and explain it in a objective way, and then culminate with the simplest interpretation of X is, and then explain it. And I think that was really important, to not overinterpret what we were seeing. And it's also important to show it in more than one way. So those sorts of things were going through my mind, figuring out how to communicate this to my colleagues in a way that would be constructive. And over the years, then, of course, there was more validation by ourselves and by others, and we got people to believe what we had initially shown. And it expanded. So it's not just step 1 and 2 now, it's step 3 and step 4 and step 5, which was very exciting to us as well. And how did we discover this? By studying human diseases. So we can go back to my initial statement of looking at the causes of human diseases. And what are the causes? Problems in how we express our genetic material. So what does the cell do? It has a way to deal with problems. So this is how we discover the quality control mechanism. And then when no mechanism is 100% efficient, so we figured out the rules, and we figured out what diseases are recessively inherited, meaning you need a defect inherited from both the mom and the father, and what are inherited in a dominant way, meaning you need the defect from only one parent to manifest disease. So it all came back then to the diseases per se. We saw this in a number of diseases. Then we figured out mechanisms getting away from the diseases. And now we're going back and helping to explain the diseases and work on therapeutics.
Very cool. Well, Lynne, congratulations to you and your laboratory members on this exciting series of discoveries. And I think it's so exciting to talk about the wonderful successes in science. But we are going to take a moment and talk about life outside of science again. And this time I want to chat with you about what you are reading. I'm always trying to squeeze in more time to read for fun, whether it's on airplanes or traveling to and from places. So do you have a favorite book, Lynne, that you can recommend for me and our listeners today that you've really enjoyed?
Well, I've read Sid Mukherjee's books on the gene and the Emperor of All Maladies, but I'm also reading a book called The Nightingale. And I love to read books about empowered women. It's about World War II in occupied France, and it's about 2 women sisters. Their father gave up on them after their mother died, and how they dealt with the war. And I love it.
I'm always looking for good books to read, so I'm going to have to add this to my list. And I've read The Gene, but I haven't yet read The Emperor of All Maladies.
Yes, and he's writing a third on immunology, so I don't know when that's going to come out, but we'll see. They're very user-friendly books, and he tells them from a personal perspective, which makes them more meaningful to us.
Absolutely. Kind of weaves that story in there, which keeps us going, so it doesn't feel like a textbook. Yeah.
Yeah.
Well, Lynne, we will add these books to our reading list for our listeners to find if they're in search of their next great read. And I know you've been doing quite a bit of traveling lately, and travel is a big part of the life of a scientist. And I think it's really important for scientists to get out there, meet their collaborators, share their research. And I'm curious, do you have a favorite place, Lynne, that science has taken you?
Oh, I love Italy. So I teach every other year. It used to be every year. Now I teach every other year at the International Center for Genetic Engineering and Biotechnology in Trieste in Italy. And I love Italy. It's a place where I can travel alone by myself. It's wonderful. I'm an adventure travel person, so I will go anywhere. I've been to the Himalaya and Vietnam, and I love Asia. I'm so excited about going to Sri Lanka.
Oh, wow. When's that coming up?
In November.
November, okay.
Well, I consult for a cancer institute in Singapore, and so before that trip, I'm going to go to Sri Lanka for a couple of days.
Very cool. So do you have your activities planned out of things that you absolutely need to see when you're there?
No, I haven't gotten there yet.
Okay. That's like part of the exciting part of travel is planning. I love it.
Yes.
So now you mentioned Italy and this teaching that you do while you're there. Can you tell us a little bit about the course of the workshop?
Sure. This is teaching largely graduate students from underprivileged countries. They come from Africa. They come from Middle East and from countries that don't have as strong a scientific history as others. For example, Hungary, some of Eastern Europe. Science is growing in Poland and Czechoslovakia. There's some very good science there, but in other countries it's not quite as strong. And South America, Argentina, there's pretty good science in Argentina, Brazil. And it's rewarding in the sense that some of the students keep in contact with me after the class. And I've helped some of the students get into programs or attend a meeting. I organized a meeting once, and in fact, I'm going to meet a student who now has her own lab who took this course, and I helped her attend a meeting I was organizing, and then she did postdoctoral work after going to graduate school and now has her own lab at the University of North Carolina. And so it's stories like these that are very rewarding for me, or being on a student's thesis committee who took the course. So I do a lot of mentoring, actually, when I travel, and I enjoy that.
That's fantastic. It sounds like it really launches their careers then, being able to participate in this course.
It's a unique opportunity for them. Yes. And we make ourselves very accessible. So we have our breakfast and our lunch with them, and we have little workshops where we meet in circles, and they can ask anything, any question, whether it pertained to science or lifestyle or whatever, so that they can figure out what they want to do with their lives.
Absolutely. Now, you mentioned this is located in Italy. Do you have a favorite thing that you like to do or a favorite place you love to see when you're in Italy?
Well, my favorite clothing designer is Italian.
That counts.
That counts, yes. Just everything, the food, the fresh air, the people. I just love walking around and just experiencing the water. Everything is just lovely. I'm pretty happy when I travel. You can put me pretty much anywhere as long as it's safe, and I'm going to find it entertaining.
Absolutely.
There aren't too many places I haven't been to.
Yeah. What's the one place that's on your bucket list aside from Sri Lanka, which you mentioned?
I've not been to Alaska, believe it or not.
Really?
So I would like to go to Alaska.
Gotcha. Do you want to do like the cruise thing or something a little different?
No.
No,
I'm not a cruise person.
Me neither.
Believe me. No. I need to get out and interact with people. And I meet most people through dogs. My husband's finally used to it.
Yeah.
If someone's walking with a dog, there's rarely a culture that is not welcoming me with open arms if I want to say hi to their dog.
Yeah. It's like people's babies, I think, in a lot of cases. So they get excited. Well, Lynne, fantastic to hear more about your travel experiences. And I think throughout our conversation, we've touched a little bit on some of the people you've been able to work with in science as well. And I think the people are part of what makes careers in science so wonderful. And I know there are stereotypes out there in the world about what scientists are like, but we do try and break those a little bit on our show by showcasing the human side of science and just some of the fun and funny things that scientists do when they're together. So do you have stories, Lynne, of any maybe quirky traditions that you experienced in a lab or just some of the fun things you've done with other researchers?
There's lots of quirky things. When I was a graduate student, we used to have the quote board. In any environment, things can be taken out of context. So as soon as someone said something that could be taken out of context, it was highly embarrassing. That went on the quote board, and you knew because it was a quote board. As soon as it came out of your mouth, oh my gosh.
It's going up, yeah.
And there's funny stories. So we used to develop things, we still do, with X-ray film, and we have a manual developer, and you have to do this in the dark room. We have red lights, but it's still pretty dark in there. And I remember, this is a long time ago, but things like this happen all the time. One of my graduate students spent a long time exposing his experiment to what he thought was X-ray film, but it was really the piece of cardboard that's on either side of the leaves of X-ray film. And
oh my gosh.
He's in the darkroom and he's developing the cardboard because he didn't really know what X-ray film looked like. And of course, you're dipping it into the developer and it totally disintegrated. So then when it came time for him to give what we call lab meeting or group meeting, everybody in the lab is like chuckling. And I'm like, okay, what's going on? And I heard the story. But there's lots of funny stories about experiences that people have in the lab that are part of the learning process.
Absolutely. I think it shows that scientists have a sense of humor too, right? We don't take everything so seriously.
We're largely just like you. I look normal, but you're right. There is a stereotype, and it's quite frustrating. So as part of the Gairdner Award, I go around and I talk to high school kids, and sometimes their parents are there at night, and I show, while everyone's sitting down, it's a PowerPoint presentation of the stereotypical scientists from movies and even TV. And it's funny in some ways, but that's really not the way we are.
Right.
Many of us. Now, do you have to have great social skills to be a scientist? One answer to that is yes, in the sense you have to be a good communicator. But other than that, no, you don't really need to. So it selects for a lot of introverts, which is fine. But scientists are really interesting people. They tend to be very well read broadly. Many are musical. A lot are very athletic. They're very interesting people.
Absolutely. And very diverse. So not necessarily the sense of race or background or things, but just in terms of interests, you're sort of multifaceted. And I think a lot of people don't necessarily appreciate that.
That's right. We're multifaceted and we tend to be very open-minded. So the issues that you read about now in terms of the general public coming to terms with certain things like gender identity, and I mean, I can speak for myself. These things have existed for centuries. These people have existed for centuries. This is sexuality and gender identification is a spectrum. And now I think that's becoming very socially, at least out in the open. I don't know how many people are buying into that, but it just makes sense from the perspective of genetics.
Absolutely. Well, it's been great to chat about some of the stereotypes about scientists and try and break some of those today. But I do want to ask you a tough question, Lynne. And this is one that I love to ask scientists because I know there are so many questions left in the world that are unanswered, but there's often things standing in the way. Things like funding, staff, technology, feasibility, the list could go on and on. But Lynne, if I gave you everything today, anything you could possibly want or ask for, what is the one question you would want to answer?
That's kind of easy. We need to figure out how to cure cancer. And this is a problem that requires we understand each individual who has cancer and each individual's cancer. And you've heard a lot or read a lot in the press probably about how people are realizing that what matters are the changes in your cells and less so the tissue that is affected by the cancer. And this is just the tip of the iceberg. This is a difficult problem. Cancer is a multifactorial problem. Cancers are not simple in terms of what's going on in the cell. That's wrong. And scientists are making very good progress on this. Thank goodness for funding. For the NIH, the National Cancer Institute is a part of the NIH, and there's a concerted large effort where basic scientists like myself who have a PhD are working with clinical people, the MDs. There are people who have PhDs and MD degrees as well, and industry are working together to make big changes in how people are being treated. We'll read a lot about that in the popular press, and certainly it's very dominant in the scientific literature. So that's the big problem, I think, that affects all of us that would be great to be solved.
Absolutely. And I think this interdisciplinary approach and this maybe collaborative approach that you mentioned is helping us get a little bit closer every day. So fantastic, big societal question to tackle, Lynne. And I'm curious, we've talked about some different aspects of your career and your work today. And I think along the course of your career as a scientist, you get a lot of wonderful advice. So is there one piece that really stands out in your mind, Lynne, that you received at some point that really helped you that you can pass on to our listeners today?
So not every day is going to be great. I think if you like what you're doing, 60 to 70% of the time, that's fabulous. There are lots of challenges in what people choose to do. It's true for science, it's true for other careers. And I think using one's time wisely, I mean, you can go to work, be there all the time and not get anything done, or you can go to work and think deeply and get something done. I think that's important. So working wisely with perseverance and realizing it's not always going to be easy and that you won't always have the best days, but that's normal and that's okay.
I like it. I think that's great advice because you're absolutely right here. Science is a process. Not every day is full of exciting discoveries and big wins. You have to trudge through some of the challenging pieces to get to those exciting discoveries. So great message to give our listeners, Lynne. Is there any other last piece of advice you want to give them, or maybe a last note of inspiration you'd like to end on?
I encourage people to understand what their weaknesses are and compensate for them. We all have weaknesses, and again, that's okay. Know your strengths and utilize them. And if you believe in something, do it despite your fears. In some ways, it's so much easier to go beyond what you think are your limits and then take a step back if you need to. And it could be a big step that's permanent, or it could be a little step, than to just stop. And I would like to encourage people to do what they have passion for. And I don't mean passion every day, and I don't mean that they're not going to have doubts about choices, but hopefully life is a long time and you want to be doing things that you feel strongly about. So choose as best as you can.
Wonderful advice, Lynne. Now, if our listeners want to learn more about you and the wonderful work you do, where should they go?
I have a website at the University of Rochester Medical Center, and I think if you Google my name, you'll get only me. So
you're quite unique, Lynne, I must say. You're easy to find.
I'm the only MAQUAT at PubMed.
Perfect. Well, listeners, definitely check out that website, see more about the projects that Lynne is working on in her lab today. And Lynne, thank you so much for joining us on our program and sharing a piece of your story.
Thank you, Marie. I've enjoyed it, and I hope I've inspired some people to think about science.
Absolutely. And listeners, thank you for joining us as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.